So I study the plasticity of the enteric nervous system, which is the nervous system of the gut. And the overall structure and function of the enteric nervous system is very much conserved across evolution. So as you can see here, the enteric nervous systems of C. elegans and Drosophila have a bunch of neurons in the foregut of the animal that control the rate of feeding. And there are some neurons that innervate the hindgut and control the rate of defecation. Now, the neurons in the foregut are unique because they form their own autonomous circuit that is completely separate from the rest of the nervous system of the animal. And the function of this circuit is to generate rhythmic contractions of the foregut or the pharynx that result in intake of food from the mouth of the animal into the intestine. So as you can see, the very rapid pumping happening over here. And I was curious to know, how does the nutritional status of the animal change the output from the foregut enteric circuit? And what I found that in fed conditions, there are about 240 pumps per minute in the animal. And if you starve the animal and put it back on food, then you see that this rate of contraction reduces by about 20%. And in cases of acute starvation that results in remodeling into the Dower stage in C. elegans, we see a complete silencing of the output from the enteric nervous system. And we see the same for the rate of defecation. If you starve animals, the rate of defecation reduces by half. And in cases of acute starvation, the rate of defecation is completely stopped. So we wanted to know what is changing the output from the enteric nervous system. And we decided to focus on the insulin signaling pathway because of its evolutionarily conserved role in responding to nutrient levels. So in favorable conditions, agonist insulin signal via the insulin receptor, which is called DAF2 in C. elegans, and via a series of phosphorylation events, it activates the kinase called AKT, which phosphorylates this transcription factor called DAF16, which is the homologue of FOXO, and retains it in the cytoplasm in an inactive state. And what happens in unfavorable conditions is that less agonists and more antagonists inhibit signaling by the insulin receptor. And as a result, we get DAF16 FOXO in the nucleus in an active form. So we wanted to know if the enteric nervous system requires active insulin signaling to maintain this rate of normal foregut contractions. So for this, I inhibited the insulin receptor only in the enteric neurons of the animal using a dominant negative strategy, where I replaced the tyrosine kinase domain of the insulin receptor DAF2 with a fluorescent protein. And I found that inhibition of the insulin receptor only in the enteric nervous system results in a significant reduction in the rate of foregut contractions of the animal. And if you starve these animals, you do not see a further reduction in their rate of foregut contractions, suggesting that reduced insulin signaling is what causes the decreased rate of feeding during acute starvation. So we wanted to know what is acting downstream of the insulin receptor, and we thought that this might be activation of the downstream transcription factor DAF16 FOXO, and we wanted to find the site of action of this transcription factor. And for this, we use the Dower stage because we see a complete silencing of the rate of foregut contractions. And we use the auxin-inducible degron or the AID strategy, where we endogenously tag the DAF16 FOXO protein with the AID tag, and we express this plant enzyme called TIR1 in a cell type specific manner. So when we add the plant hormone auxin, then auxin acts as the ligand for TIR1, and it would deplete the AID tagged DAF16 FOXO protein only from the cell type of interest. So first I created a strain that expresses the TIR1 enzyme only in the enteric nervous system of the animal, as you can see over here. And when you add auxin, you see that DAF16 FOXO is gone specifically from the enteric neurons and not from the other neurons of the body. And what we see that if you remove DAF16 FOXO only from the enteric nervous system of the animal, this is sufficient to reverse the completely silent state of foregut contractions that we see in acute starvation conditions in the Dower stage, suggesting that DAF16 FOXO activity is required cell autonomously in the enteric neurons to completely silence the output from the circuit during starvation. But what we also found that if you remove DAF16 FOXO from another organ, which is the intestine or the midgut epithelial cells, we also see a reversal of the silent state of the foregut, suggesting that there is some sort of an interorgan signal that is originating from the intestine in a DAF16 FOXO dependent manner that is communicated to the foregut of the animal to change the output from this circuit during starvation condition. And to identify this interorgan signal, I used a transcriptomic approach where I profiled the gene expression changes that occur when you remove TAF16-FOXO only from the intestine of the animal during acute starvation. And we found about 1,200 genes that were differentially expressed, with most of these genes functioning in the digestive processes, such as digestive enzymes, etc. But we found a specific family of genes that were affected by DAF16-FOXO during starvation. And these are the genes that are predicted to function in the intestinal neuropeptide secretion machinery. So these are the AEX genes. AEX1 is a snare regulator, AEX4 is a SNAP23 like T snare protein, and AEX5 is a proprotein convertase. And you can see here that during acute starvation, the expression of all the AEX genes is strongly downregulated. And if we remove TAF16-FOXO from the intestine of the animal, then we see that the AEX gene expression goes up, with AEX1 showing the strongest change in both directions. So very little is known about the ability of epithelial cells of the gut in terms of releasing neuropeptides. So we wanted to know if this is something specific for the C. elegans gut, or do we also see this in other organisms? So I looked at a recently published single-cell RNA-seq data set that profiled the different cell types in the mammalian gut. And I found that the homologs of the three AEX genes show very strong expression in the EC cells, which are the enterocytes, or the most abundant epithelial cell type in the villi of the small intestine of mammals. So because the expression of AEX genes reduces during starvation, I wanted to test this by creating CRISPR-based endogenous GFP reporters. And here I'm showing you the expression pattern of AEX1. As you can see that AEX1 shows very strong expression in the intestinal cells of the animal, both in the adult stage as well as in the larval stages, but only in the fed conditions. Because during cases of acute starvation, as in the Dower stage, you see this expression is completely gone. So because AEX1 is predicted to function in the secretion machinery for neuropeptides, I wanted to test this directly by creating a secretion reporter. So I used an intestine-specific promoter called GES1, and I drove the expression of an insulin peptide called INS1 that is translationally fused to tag RFP. And then I have SL2-GFP-H2B, where SL2 is a trans-splicing sequence that would split this into two separate mRNAs, where the GFP-H2B goes to the nucleus and reports on the transcription of this cassette, and the tagged insulin peptide would be released from the intestinal cells, and it accumulates in the scavenger-like cells called coelomocytes in the body cavity. And as you can see that in starved animals, the ability of the intestinal epithelial cells to release this insulin peptide is significantly reduced. Now, is this because these animals have reduced AEX1 expression? And to test this, I created a loss of function allele for AEX1 using CRISPR-Cas9. And what I found that if you remove AEX1, the ability of intestinal cells to release insulin peptides is significantly reduced even in the abundance of food, which led us to this model that it might be affecting foregut contractions as well. So we looked at the rate of foregut contractions in multiple AEX loss of function mutants, and we found that these animals has a reduced rate of foregut contractions even in the abundance of food. So based on these data, we proposed that in well-fed conditions, the intestine releases some interorgan signal that acts on the insulin receptor in the enteric neurons of the foregut that results in normal rate of pumping. And during starvation conditions, DAF16-FOXO gets activated and it shuts down the secretion machinery. And in the absence of the interorgan signal, we see reduced output from the enteric nervous system. So to identify this interorgan signal, we looked at a recently published single-cell data set, and we found that there are only two insulin peptides, INS7 and INS35, that are specific for the intestinal cluster. And I validated this by making CRISPR-based GFP reporters for both of these insulins. And what we also found that if you remove these insulin peptides, then removal of either INS7 or INS35 results in a significant reduction in the rate of foregut contractions, even in the abundance of food. And if you remove both of them together, you do not see an additive effect, suggesting that they are acting in an epistatic manner. And these insulin peptides are not only necessary for the normal rate of foregut contractions, but if you constitutively express them from the intestinal epithelial cells during acute starvation conditions, then you see that this is sufficient to reverse the silent state of the foregut, suggesting that these are not only necessary for the normal rate of feeding in nutrient abundant conditions, but also sufficient to reverse the silent state of the foregut during acute starvation conditions. Now, we wanted to know where are these insulin peptides released by the intestinal epithelial cells received within the enteric circuit. So I focused on multiple neurons in the enteric circuit that are part of either the pumping module or the neuromodulation module. And one of the neurons stood out, which is the NSM neuron, which is a serotonergic neuron in the foregut of the animal. And what I found is that if you inhibit the insulin receptor, specifically in the serotonergic NSM neurons, using the dominant negative insulin receptor strategy that I talked about earlier, you see a reduction in the rate of foregut contractions, even in the presence of food. And if you starve these animals, you see that the effect of starvation on feeding behavior that you see in wild type animals is strongly suppressed. So is NSM the neuron that is receiving the insulin signals from the gut? And for this, I deleted IN7 and INS35 in a background where the insulin receptor was already inhibited in the NSM neurons of the foregut. And what I found is that this did not cause a further reduction in the rate of foregut contractions, suggesting that the NSM neuron is the primary site within the enteric circuit that is receiving these interorgan signals from the midgut epithelial cells to change the output from the enteric nervous system. So just to sum up what I showed you today, what I showed is in favorable conditions, food cues are sensed by sensory neurons and they release agonist insulins to prevent the activation of the transcription factor DAF16 FOXO. And the midgut epithelial cells release two distinct classes of neuropeptides. We talked about the insulin peptides IN7 and INS35 that act on the foregut enteric neurons NSM to sustain the normal rate of feeding. And I did not have time to talk about the other family of neuropeptide, which is the non-insulin neuropeptide NLP40 that is also released from the midgut epithelial cells and acts on the hindgut enteric neurons AVL and DVV to sustain normal rate of defecation. And what you see in acute starvation conditions that in the absence of food, the transcription factor DAP16 FOXO gets activated in the midgut epithelial cells, and this shuts down the secretion machinery of the intestine via transcriptional silencing of the AEX genes. And in the absence of these two peptidergic signaling axes from the midgut to the enteric nervous system, we see reduced feeding as well as reduced defecation. So the intestine is acting as the integrator of the nutritional status of the animal. And based on the availability of food, it is changing the output from two different circuits of the enteric nervous system to regulate feeding and defecation behavior by modulating different neuropeptidergic signaling axes. So with this, I would like to thank my advisor, Oliver, members of the Hobart Lab, people who provided the data, resources, and strains, and HHMI for funding. And I'm happy to take any questions.
Great, thank you very much. A virtual round of applause for you. If you have any questions, please type them into the Q&A box and I will read them out on your behalf. To get going, you kind of answered this question in my last slide, but I'll ask it anyway. So you mentioned that there's a sort of standalone circuit for the foregut. What controls peristalsis in the rest of the gut in the worm? Is there separate circuits for each section of the gut?
That's a great point. So in the midgut is directly not innervated by any neurons, but the midgut epithelial cells seem to have a pacemaker where they produce this rhythmic calcium waves, and this calcium wave results in release of the non-insulin neuropeptide called NLP40 that I talked about today that acts on the hindgut enteric neurons AVL and DVB to promote defecation. So this is something that some sort of a signal in the gut that is triggered by presence of food in the posterior gut triggers this defecation behavior, which you do not see when the animal is not actively eating. But the foregut enteric circuit is a completely autonomous circuit that is can function even when the animal is not actively eating as such.
Okay, thank you. And you mentioned that there are food cues that initiate this signaling of insulin peptides. Do you have any idea whether what those cues are, whether specific diets that are more strongly activating food cues than other diets? Could you induce a DAWA-like response even though the animals are still eating because they're, lacking a certain nutrient, for example?
That's a great point. So recently, Steve Flavel's lab at MIT has shown that it is specific parts of the bacterial cell wall and the membrane that triggers this feeding response.
Mhm.
Uh, but what they found is that the increase in the rate of foregut contractions that we see when the animal is eating food is something that is triggered by stuff that is on the outside of the bacteria.
Mhm.
And It is present in most bacterial species, but if the bacteria is pathogenic, then it is inhibited via different pathways. So there is some sort of computation happening there. We have identified some of the molecules, but we definitely, in the natural environment, C. elegans is exposed to many different species of bacteria, and we don't know the full ecological context of these findings.
Sure. If I have one last question, if there's no in the Q none in the Q&A box. Um, so presumably the induction and loss of this signal varies quite a lot depending on the food and the time of day that the animals are eating. But presumably the contractions are quite consistent and there's a rhythmic contraction that occurs at a relatively constant rate. So how is timing encoded in this signaling network?
In terms of in terms of the diurnal variations of the
In
pumping,
terms
or
of, yeah, in terms of, if a worm has eaten, it's created the cue to create these contractions, but then that cue is no longer there, but the contractions are still happening. Does that occur?
I see.
And what controls the sort of on and off switch of those contractions?
So one thing I did not mention is that C. elegans, because it has a very short reproductive period, it makes
Hmm.
all its 300 babies in the first 3 days of its adulthood. So
Right.
there's an extreme demand for nutrients. So during this period, it constantly pumps. It doesn't
Okay.
stop. If you take food away, then after a few hours you start seeing a reduction. But I forgot to mention that even though I'm measuring the effect of starvation, all the recording is done after the starvation when the animal is put back on food. So
Hmm.
what I'm checking is not the presence or absence of food, but the internal state of the animal in terms of
Sure.
its feeding status that is determining this. So as you mentioned that there are multiple factors that regulate this rate. The fact of that the animal is actively eating um, activates the serotonergic neuron that releases serotonin to promote the rate of pumping. Other than that, the pacemaker of the circuit, which is a cholinergic neuron, it releases acetylcholine and causes contractions. And that rate of the pacemaker remains more or less constant,
Okay.
independent of whether the active animal is actively eating or not. And this slows down during aging after the animal has completed reproduction. But overall, in early days of adulthood, it remains very consistent.
Okay, great. Thank you. That's very, very helpful to clarify. Thank you.